CSIR Evolution Chapter 10. Chapter 10. Assessment of Animal Behavior. The term ethology derives from the Greek language ethos meaning character and logia meaning the study of. Ethology involves study of animal behavior in natural surroundings. Behavior can be defined as the way an organism responds to stimuli in its environment in order to survive and reproduce. A stimulus is always a change in the internal or external environment of organisms. The stimulus may be in the form of signs/symbols or releases. Signs can be visual, auditory, chemical, or other types of sensory models. Release or stimulus releases certain programmed stereotyped activities or fixed action pattern, FAP, of behavior. The stimulus always elicits a response, which is an attempt to adapt to the change. Thus, behavior is generally stimulus-response oriented. Stimuli might be as simple as the odor of food. In this sense, a bacterial cell behaves by moving toward higher concentrations of sugar. This behavior is very simple and well suited to the life of bacteria, allowing these organisms to live and reproduce. As animals evolved, they occupied different environments and faced diverse problems that affected their survival and reproduction. The behavior of an animal is essentially an expression of the capabilities of its nervous system. Nervous systems perceive and process information concerning environmental stimuli and trigger adaptive motor responses. While the environment determines the nature of the stimulus, the response involves the body, especially the nervous system of the organism, which is genetically determined. We define animal behavior as any internally coordinated, externally visible pattern of activity that responds to changing external or internal conditions. Internally coordinated refers to internal information processing such as endocrine signaling, sensory information processing, or the action of neurotransmitters. When two male giraffes meet during the breeding season, such processes coordinate their aggressive behavior. Externally visible activity refers to patterns that we can observe and measure. For example, we can observe a squirrel eating a nut and can quantify this behavior. We can, however, observe an animal's behavioral response to changing conditions. For example, male crickets, frogs, and birds vocalize in response to changes in day length, temperature, or moisture at specific times of the year. Behavioral studies are of great importance in increasing our understanding and appreciation of non-human animals. In addition to providing knowledge about the diversity and complexity of behavior in nature, such studies also provide information crucial to improvements in the welfare of animals maintained in laboratories, agricultural settings, and zoos, and as companion animals. As animals ourselves, animal research provides a window into our own behaviors. 10.1 History of Animal Behavior. The study and interpretation of the natural behavior of animals is a fascinating subject, and those who choose to study animal behavior are concerned with understanding the causes, functions, and evolution of behavior. The overall study of the behavior of animals in their natural environment has a long history because human beings were hunter-gatherers. It was thus compulsory for them to be careful observer of animal behavior, and in fact revealed in the paintings on the walls of excavated caves of prehistoric times. The great Greek philosopher Aristotle, 372 BC, had many interesting observations concerning animal behavior. He wrote excellent description of animal behavior in his book Historia Animalium, meaning the history of animals. He clearly recorded the various behaviors of animals. However, the inferences he drew from his observations were incorrect. Another keen observer of animal behavior was William Harvey, a 17th-century scientist who studied the breeding, nesting, and incubation behavior of many birds. Gilbert White, 1720 to 1793, a preacher in England, made careful observations of behavioral aspects of man and animals, especially the swallows. Like Aristotle, he was unable to interpret his finding by using a scientific approach. He believed that swallows bury themselves in the muddy bottom during winter. He arrived at this conclusion as the swallows aggregates in the reef beds before they migrate during the night. Ethology was directly influenced by the works of Charles Darwin, 1809 to 1889. He is regarded as the first one to make scientific study of animal behavior in his book, The Expression of Emotions in Man and Animals. The great number of facts he collected about the behavior of Animals in their natural environment made it possible to distinguish the basic categories of behavior, namely instinct, learning ability, and the elementary ability to reason. Darwin also showed that animals' behavioral traits, like physical traits, are inherited and subject to change. Using instincts as an example, Darwin indicated the ways in which behavioral traits can evolve in the process of natural selection. Charles Darwin, in his classic book 1859, On the Theory of Evolution by Natural Selection, devoted a whole chapter to what he called instinct. As early as 1873, the British amateur investigator Douglas Spalding recorded some very interesting observations on the attachment behavior of young domestic chicks to abnormal objects, a phenomenon that was later called imprinting. Charles Otis Whitman, 1842 to 1910, who coined the term instinct to describe the display patterns of pigeon. Wallace Craig, 1876 to 1954, was an American ethologist and animal psychologist. He provided a conceptual framework for the study of behavior organization. Craig experimentally studied the behavioral expression of emotion, the way innate and learned behavioral tendencies are integrated, and how vocal as well as social behaviors are organized. He encouraged a view of behavior as an integrated process with evolutionary, motivational, experiential, social, and ecological degrees of freedom. This integrative perspective helped shape modern behavioral scene. Jacques Loeb, 1859 to 1924, was a German-born American physiologist and biologist. Loeb developed a theory of animal behavior based on the concept of tropism, an involuntary forced movement. He found that an animal's response is a direct and autonomic function of a reaction to a stimulus. In other words, behavior is said to be forced by a stimulus. It does not require any explanation in terms of an animal's consciousness. Loeb's theory was very influential for the time of biological sciences, which represented a change from the work of Romanes and Morgan. H.S. Jennings, 1868 to 1947, demonstrated by his works that animals are capable of performing orientation movements learned by trial and error. Ivan Petrovich Pavlov, 1849 to 1936, a Russian ethologist, elaborated the classical conditioning theory. He carried out his classical experiments on salivary gland secretions and the role they play in digesting food, for which he eventually won the Nobel Prize. Pavlov noticed that dogs would often salivate profusely just prior to being fed and discovered that this behavior could also be produced in the absence of food if he first paired the presentation of food with a novel stimulus, like the ringing of a bell. After several such pairings, dogs associated the novel stimulus with food and would salivate after hearing it, a behavior he called a conditional reflex. Classical or Pavlovian conditioning occurs whenever a novel stimulus, like the ringing of a bell, is paired with an existing stimulus, here the food, and elicits a particular response, salivation. Eventually, the novel stimulus alone elicits the same response as the existing stimulus. E.L. Thorndike, 1874 to 1949, was a pioneer not only in behaviorism and in studying learning, but also in using animals in psychology experiments. Thorndike was able to create a theory of learning based on his research with animals. His doctoral dissertation, Animal Intelligence, an experimental study of the associative processes in animals, was the first in psychology where the subjects were non-humans. He was interested in whether animals could learn tasks through imitation or observation. Oscar Heinroth, 1871 to 1945, was a German biologist who was one of the first to apply the methods of comparative morphology to animal behavior, and was thus one of the founders of ethology. His extensive studies of behavior in the Anatidae, ducks and geese, showed that instinctive behavior patterns correlated with taxonomic relationships determined on the basis of morphological features. He also rediscovered the phenomenon of imprinting, reported in the 19th century by Douglas Spalding. His results were popularized by his student, Konrad Lorenz. Lorenz regarded Heinroth as the true founder of the study of animal behavior seen as a branch of zoology. The modern discipline of ethology is generally considered to have begun during the 1930s with the work of Dutch biologist Nicolaas Tinbergen and by Austrian biologists Konrad Lorenz and Karl von Frisch, joint winners of the 1972-73, the Nobel Prize in Physiology or Medicine 1973, for their discoveries concerning organization and elicitation of individual and social behavior patterns. They are also credited for introducing an absolutely new branch of science to zoology and making it popular among common people. Karl R. Ritter von Frisch, 1886–1982, was a German-Austrian ethologist. His work centered on investigations of the sensory perceptions of the honey bee. He was one of the first to translate the meaning of the waggle dance. His theory, described in his 1927 book Aus dem Leben der Bienen, translated into English as the dancing bees, was disputed by other scientists and greeted with skepticism at the time. Only much later was it definitively proved to be an accurate theoretical analysis. Konrad Zacharias Lorenz, 1903–1989, was an Austrian zoologist, ethologist, and ornithologist and considered to be one of the principal founders and father of ethology. With Oscar Heinroth, he is held to have discovered imprinting, an especially rapid and relatively irreversible learning process that occurs early in the individual's life where auditory and visual stimuli from an animal's parents are needed to induce the young to follow their parents. Central concept complementary to imprinting is the innate release mechanism, whereby organisms are genetically predisposed to be especially responsive to certain stimuli, such that imprinting will become fixed on the parents. He formulated imprinting behavior in Gosling's, his books, King Solomon's Ring and Man Meets Dog, demonstrates his deep understanding on animal behavior. Nicolaas Nico Tinbergen, 1907–1988, was a Dutch biologist and ornithologist. In 1951, he published The Study of Instinct, an influential book on animal behavior. He worked on the courtship behavior in stickleback fish, food begging in chicks of seagulls, nesting behavior of digger wasps. Burrhus Frederick Skinner, 1904–1990, was an American psychologist, did extensive work on learning by reward or reinforced learning in caged rats. He developed novel tools and procedures to study the development of behavior. Most famously, he is known for the creation of operant chambers or Skinner boxes, which house a single animal. Skinner examined how the behavior of animals could be modified by pairing a particular behavior with either a reward or a punishment. Behaviors could be enhanced if animals were given food rewards, a positive reinforcement, for performing the behavior. Alternatively, these behaviors could be reduced if they resulted in a painful shock, positive punishment. In this process, called operant conditioning, an animal learns to associate a behavior with a particular consequence, either positive or negative. Harry Frederick Harlow, 1905–1981, was an American psychologist who provided a new understanding of human behavior and development through studies of social behavior of monkeys. His research contributions in the areas of learning, motivation, and affection have major relevance for general and child psychology. He is best known for his studies on maternal separation and social isolation, experiments on rhesus monkeys, which demonstrated the importance of caregiving and companionship in social and cognitive development. Donald Griffin, 1915–2003, is credited with the development of cognitive ethology, animal navigation, and echolocation in bats. He published book The Question of Animal Awareness. Today, cognitive ethologists study topics such as learning and memory, counting ability, symbol recognition, and vocal communication in organisms as diverse as bees, crows, rodents, and primates. Richard Dawkins, born in 1941, he is a British ethologist and evolutionary biologist. Dawkins came to prominence with his 1976 book, The Selfish Gene, which popularized the gene-setter view of evolution and introduced the term meme, the behavioral equivalent of a gene. In 1982, he introduced into evolutionary biology the influential concept that the phenotypic effects of a gene are not necessarily limited to an organism's body, but can stretch far into the environment, including the bodies of other organisms. This concept is presented in his book, The Extended Phenotype. In his 1986 book, The Blind Watchmaker, he argues against the existence of a supernatural creator based upon the complexity of living organisms. Instead, he describes evolutionary processes. Edward Osborne Wilson, born 1929, usually cited as E.O. Wilson, is an American biologist. Wilson is an influential biologist who, on numerous occasions, he has been nicknamed the New Darwin, or the Darwin of the 21st century. His biological specialty is myrmecology, the study of ants, on which he has been called the world's leading expert. Wilson has been called the father of sociobiology and the father of biodiversity. Among his greatest contributions to ecological theory is the theory of island biogeography, which he developed in collaboration with the mathematical ecologist Robert MacArthur. Wilson has argued that the unit of selection is a gene, the basic element of heredity. The target of selection is normally the individual who carries an ensemble of genes of certain kinds. With regard to the use of kin selection in explaining the behavior of eusocial insects, the new view that I'm proposing is that it was group selection all along, an idea first roughly formulated by Darwin. Sociobiological research was at the time particularly controversial with regard to its application to humans. The theory established a scientific argument for rejecting the common doctrine of tabula rasa, which holds that human beings are born without any innate mental content and that culture functions to increase human knowledge and aid in survival and success. Desmond John Morris, born 1928, is an English zoologist, ethologist, and surrealist painter, as well as a popular author in human sociobiology. He is known for his 1967 book The Naked Ape and for his television programs such as Zoo Time. He studied the picture-making abilities of apes. Clive D. L. Wynne, born 1961, is a British-Australian ethologist specializing in the behavior of dogs and their wild relatives. Wynne's earlier research focused on the cognitive abilities of pigeons, as well as their perception of arbitrary short-time intervals. Jeffrey C. Hall, Michael Rosbash, and Michael W. Young worked on molecular mechanism of biological RHYTMS. Using Drosophila as a model organism, they isolated a gene that controls the normal daily biological rhythm. They showed that this gene encodes a protein that accumulates in the cell during the night and is then degraded during the day. They were awarded with Nobel Prize in Physiology or Medicine in 2017 for their discoveries of molecular mechanisms controlling the circadian rhythm. 10.2 Methods to study behavior. The main challenge for study of the behavior of animals is to understand the fundamental nature of the scientific approach that must be used more formally. Only a laborious experimental characterization of behavioral phenomena will yield common generalities on how and why animals do what they do, rather than providing a simple catalog of stories. We must be able to quantify our behavioral observations with numbers according to specific rules, and different people must be able to recognize an animal behavior independently. Often, such characterization begins when a researcher completes an ethogram, which is a formal description or inventory of an animal's behaviors. Ethograms typically list or catalog defined, discrete behaviors that a particular species exhibits. Researchers can use an ethogram and measure its frequency, how many times a behavior occurs, duration, the length of time of a behavior, rate, the frequency of the behavior per unit time, or intensity, the vigor or forcefulness of the behavior. Sometimes only certain behaviors, such as social behaviors, e.g., grooming self, grooming others, chasing others, are quantified. Typically, a more complete list of behaviors that occur over a specific time period is recorded, e.g., sleeping, grooming self, grooming others, walking, climbing tree, eating food, drinking. From these observations, a researcher can then determine both the total and the relative time that an animal engaged in each behavior. In other words, the measure of the behavior divided by the overall time spent observing the animal. The resulting time budget indicates the total time and relative frequency of each behavior. Ethograms are commonly constructed for animals in captivity and can be used as a baseline for healthy behavior. Scientists use the scientific method to learn about the natural world. Figure 91. The scientific method is a formalized process that involves the testing of hypotheses. Researchers analyze data to determine whether they support or refute their hypothesis. Science never proves hypothesis is correct. Rather, it is an ongoing process of hypothesis testing, and that process is crucial to scientific progress. A hypothesis that makes broad predictions and stands up to repeated testing may eventually become a theory. Scientists must consider the limitations of studies that report correlations, because correlation does not demonstrate causation. Figure 91. The scientific method. Animal behavior researchers generate and test hypotheses. Hypotheses can be developed through observations or through the use of analytical models. To test hypotheses, Researchers commonly use observational, experimental, and comparative methods. 10.2.1 Observational method, an approach in which scientists observe and record the behavior of an organism without manipulating the environment or the animal. It is least invasive to the animal and provides data both to describe behavior and to test hypotheses. For example, the observational method is used to construct ethograms. Altman, 1974, proposed different animal sampling techniques for observational methods. 1. Ad libitum sampling. Ad libitum in Latin means at one's pleasure. A group of animals is selected and the observer remains with this group for a considerable period of time to observe all activities of the group and records any behavior of any individuals that appears relevant to the observer. No constraints are placed as to what should be recorded and when. It is often used during the initial phase of descriptive studies and during reconnaissance observations when formulating and fine-tuning research questions. Although measures of frequency and duration are sometimes made during ad libitum sampling, they are gathered in a haphazard manner so that they represent only pilot or first approximation data. 2. Sociometric matrix completion. In some studies, ad libitum sampling has been supplemented by making additional observations on particular pairs of individuals. This has been accomplished by spending more time with these individuals, or e.g., by experimentally provoking a fight by means of competition in pairs of individuals for whom the original sample size was considered inadequate. The results of such sampling are usually published in the form of a sociometric matrix, that is, contingency table in which actors, e.g., aggressors or winners, are represented by the rows, and recipients, e.g., losers of fights, by the columns, and in which the cell entries indicate the frequencies of the corresponding dyadic interactions. In these studies, the object has been to establish, for each pair, the direction and degree of one-sidedness of some relationship, such as groomer-groomee or winner-loser of fights. In such studies, then, a sample usually is considered to be inadequate if the data in a pair of cells are small in number or large in number but nearly equal. 3. Focal animal sampling. It provides unbiased data from different individuals. In this method, a focal individual is randomly selected and observed for a specified time period, and all pertinent behaviors performed by the individual are recorded. After the time period has expired, a different individual is randomly selected and observed. Focal animal sampling has several advantages. 1. The behavior of different individuals is sampled. If individuals have unique markings or morphological aspects, the researcher can record the behavior of known individuals and identify the total number of unique individuals sampled. If it is difficult to identify individuals, the researcher can move to a different location before selecting a new individual. The researcher can also simply select an individual that is some distance away from the last focal animal observed. This ensures that a number of different individuals are sampled. 2. Because random individuals are selected, instead of individuals that exhibit either unusually high or low levels of a particular behavior, sampling is unbiased. 3. Because data are collected over a recorded amount of time, researchers can easily calculate the frequency of a behavior and the rate at which the behavior was displayed. 4. This technique requires little more than being able to keep track of time and observe and record behavior. Depending on the situation, these tasks might best be accomplished by a pair of researchers, a recorder who keeps track of time and records the behavior, and an observer who watches the animal and describes the behaviors displayed. 4. All occurrence sampling. Under some conditions, it is possible to record all occurrences of certain classes of behaviors in all members of the group during each observation period. There are certain behaviors are performed by several animals at the same time. For example, one animal starts the alarm call and other animals follow. In such cases, the behavior can be recorded as one event. One can record the number of alarm calls per unit of time, which will provide rate of occurrence of the behavior in a fixed period of time. Such records are generally possible only if observational conditions are excellent, if the behaviors are sufficiently attention-attracting that all cases will be observed, and in the behavioral events never occur too frequently to record. With a wise choice of sample periods, all occurrence sampling can provide accurate information about the rate of occurrence in temporal changes in the rate of such behavior in the group as a whole. Although this is not the technique of choice for many kinds of sequential analysis. However, if the sequential information that is desired is that sequelae of some behavior that can be sampled in this way, e.g. the response to vocal threats, one could start each sample with an observation on such behavior, then record what happens next. This sampling technique is appropriate for studies of behavioral synchrony if the observational and recording conditions are such that occurrences of the behavior can be recorded even if they are simultaneous. 5. Sequence sampling. In sequence sampling, the focus of observation is an interaction sequence, rather than any particular individuals. A sample period begins when an interaction begins. During the sample, all behaviors under study are recorded in order of occurrence. The sample continues until the interaction sequence terminates or is interrupted, and the next sample begins with the onset of another sequence of interactions. 4. Example, sequence sampling has been used in studies of social behavior in crabs and monkeys. 6.10 Sampling. The observation of everyday behavior of an individual or a group of individuals for definite short periods of time, and the recording of the occurrence, yes or non-occurrence, no, of certain specified and objectively defined forms of behavior during each of these periods. 4. Example, attack behavior in territorial animals or infant killing in monkeys. The common features of the technique are the following. 1. In each sample period, occurrence or non-occurrence, rather than frequency, is scored. 2. Interactions of just a single individual or pair of individuals are recorded in each sample period. 3. Occurrence, for most users of this technique, is meant in process at any time during the sample period, i.e. sampling of states rather than events. 4. The sample periods are usually short, e.g. 15 seconds, with about 20 sample periods in succession. Such batches of samples may then be repeated, perhaps twice a day over the period of the study. 1. Instantaneous for scan sampling. Instantaneous sampling is a technique in which the observer records an individual's current activity at pre-selected moments in time, e.g. every minute on the minute throughout the day. Here, data are collected from individuals at regular time intervals. It is a sample of states, not events. For instance, you might be interested in the frequency with which individuals in a group scan their environment. I raise their head to observe their surroundings. You could use scan sampling at 30-second intervals to record the number of group members whose head is raised at a given moment. Over a sufficiently long observation period, these data would provide information on the percentage of time individuals spend scanning the environment. An advantage of observational method is that researchers can observe naturally occurring behaviors. However, many factors might influence the behavior observed, and so it is difficult to draw firm conclusions about why particular behavior patterns occur from observations alone. To do that, we need a different method that provides greater control. 16.2.2 Experimental method. An approach in which scientists manipulate or change a variable to examine how it affects the behavior of an animal. The variable that is changed is called the independent variable, and it can be anything that we measure, control, or manipulate. It can be abiotic, e.g., temperature, humidity, wind, or biotic, e.g., habitat, food availability, social interactions. The researcher then measures changes in another variable, the dependent variable, that occur in response to changes in the independent variable. For example, suppose you were examining how food availability affects clutch size, the number of eggs laid by a female during one reproductive attempt. Your experimental treatment might consist of manipulating the amount of food, the independent variable, and then measuring the number of eggs produced, the dependent variable. In order to determine whether or not your manipulation is actually influencing the dependent variable, it is essential to include a control group. The control group does not experience the manipulation, but is treated similarly in all other aspects. In the simplest experimental design, only one factor differs between the experimental and control groups. In essence, the control group represents the null hypothesis, and the experimental group represents the alternate hypothesis. 10.2.3 Comparative method. An approach that examined differences and similarities between species to understand the evolution of behavioral traits. It is often observed that sets of closely related species share similar behavioral adaptations because their shared ancestor possessed the behavioral trait. By comparing the behavior of closely related species, researchers can try to understand whether a behavior is ancestral or derived. Ancestral trait is found in the common ancestor of two or more species. It is also called a plesiomorphic trait. Derived trait is found in an organism that was not present in the last common ancestor of a group of two or more species. It is also known as an apomorphic trait. To understand the evolution of behavioral traits, researchers often require knowledge of the evolutionary relationships among taxa. Taxonomic groups, such as species. Evolutionary relationships are provided by a phylogeny, hypothesized evolutionary history of a group. A species. For any set of species, a phylogeny illustrates our best understanding of the patterns of descent, showing ancestor-descendant relationships that reflect their evolutionary history. For example, two species that are more closely related to one another than to any other. Species share a recent common ancestor and are known as sister species. In a phylogenetic diagram, these species are represented by two lines that share a node, the common ancestor. 16.2.4 Laboratory methods. Some of laboratory techniques which can be used to assess animal behavior are 1. Neuroanatomical or lesion or ablation techniques. We know that different types of behavior are controlled by specific regions of the brain. Doctors observed that patients showed peculiar behavioral changes due to injury in head. It was rightly correlated by them that behavior is controlled by brain. While performing the operations or post-mortem, autopsies, the destructed areas of brain were identified, and impairment in behavior was correlated with it, indicating direct influence of brain over behavior. The process of identifying the parts of the brain that are involved in language began in 1861, when Paul Broca, a French neurosurgeon, examined the brain of a recently deceased patient who had an unusual disorder in left frontal lobe. The person with damaged area is able to understand spoken language and did not have any motor impairments of the mouth or tongue that might have affected his ability to speak. He could neither speak a complete sentence nor express his thoughts in writing. This area responsible for speech production and writing has been named as Broca's area. It is found in the left inferior frontal cortex lobe. Karl Wernicke, a German neurologist, discovered another part of the brain, termed as Wernicke area, which is involved in understanding language in the posterior superior portion of the left temporal lobe. People who had a lesion at this location could speak, but their speech was often incoherent and made no sense. Wernicke's area is primarily involved in the comprehension. Historically, this area has been associated with language understanding, whether it is written or spoken. It is also a storehouse of vocabulary. Broca's area and Wernicke's area are connected by a large bundle of nerve fibers called the arcuate fasciculus, figure 92. Neuroanatomical methods are the oldest and crude method for studying the relationship between particular neural structure and behavior. In this method, certain regions of the brain were destroyed and their function was deduced from the abnormal behavior it caused in the animal. The larger ablation, removal of tissue, or lesion, pathological changes in tissue, were carried out freehand by knife cuts, whereas the localized and small lesions are produced by passing electric current through platinum iridium electrode to cauterize the area. Carl Lashley, 1938, conducted his studies on memory by ablation on different brain parts of rats which were trained to running maze. Stereotaxic equipment can be used to place small and precise injuries in brain by making cuts with a knife or by the neurotoxic kyanic acid. And behavior is observed. A stereotaxic device uses a set of 3 coordinates that, when the head is in a fixed position, allows for the precise location of brain sections. This technique helped many earlier workers to form brain atlases or brain maps, which are known as stereotaxic atlases that are presently available from many mammalian species vis. On cats by Jasper and Marson, 1954, on rats by de Groot, 1959, and on dogs by Lin and co-workers, 1961. When rats' nucleus accumbens is stereotaxically ablated, the increase in aggressive behavior and decrease in maternal behavior was observed. The nucleus accumbens is a region in the basal forebrain rostral to the preoptic area of the hypothalamus. It is an aggregate of neurons which is described as having an outer shell and an inner core. The nucleus. Accumbens, being one part of the reward system, plays an important role in processing rewarding stimuli, reinforcing stimuli, e.g. food and water, and those which are both rewarding and reinforcing. It is believed to play an important role in drug addiction. Nowadays, for ablation, laser lights are used, which is capable of making very precise and minutest ablations in desired region of brain. 2. Neurophysiological techniques. Neurophysiological methods are based on the discoveries that messages in brain are passed on using electrical current and chemicals, called neurotransmitters. The bioelectricity was discovered by chance. The Italian scientist Galvani, 1786, hung some frog legs on his balcony railing one day and noticed that the legs twitched when they touched the metal fence. This finding laid the foundation for electrophysiology. The discovery that the messages in nervous tissue travel in the form of electric current led to a less destructive and more precise neurophysiological method to study behavior. David Ferrier, 1843–1928, was able to roughly localize in the cerebral cortex the sensory centers for vision, auditory, gustatory, and olfactory sensations. It became technically possible to record the electrical potential of active nerves with the aid of implanted electrodes and to amplify this activity by a highly sensitive instrument, where it could be detected and measured. This technique has now become a routine tool of neurophysiologists and neuroethologists. The procedure involves implanting electrodes by stereotactic surgery in the brain and recording of electrical activities from brain while animal is behaving. At present, physiological studies can be done by recording electrical activity of brain by EEG or by stimulating different areas of brain by planting electrodes. Alpha, beta, theta, and delta waves are recorded by EEG. Alpha waves that are believed to emanate from the parietal and occipital lobes of brain reveal resting and peaceful relaxed state of brain that is otherwise alert. Beta waves are produced in frontal lobes and indicate the daily mental activity, concentration, and thought. Theta waves denote emotional stress and sometimes hallucination. Delta waves are generated in deep sleep. 3. Neurochemical techniques. Neurons are affected in their activity by local application of neurotransmitters. Microcannula fixed by stereotaxic apparatus can be used to inject minute quantities of chemicals in precise locations of brain, such as limbic system, and behavior can be recorded. Neurochemical techniques involve stimulation of parts of brain by drugs such as alcohol, opium, hashish, bang, etc., which alter the behavior of the animal. For example, if a researcher intends to stimulate neurons via glutamate receptors in medial septum of an experimental animal, he will use stereotaxic apparatus, slow perfusion pump, and microcannula used to administer injection treatments. Tranquilizers, barbiturates, and drugs like compose, larpose, etc. are psychoactive drugs which affect the brain and change the behavior of animals. Hormones such as estrogen and testosterone can be introduced into hypothalamus through cannulation, and the behavior changes can be recorded. Adrenaline, histamine, testosterone, and dopamine stimulate different parts of the limbic system. Fisher, 1956, showed that sexual and maternal behavior can be induced by the injection of minute quantities of sex hormones in the regions of hypothalamus. Stimulation of amygdala brings about aggressive behavior, and stimulation of septum pellucidum gives immense pleasure to the animal. At present, advanced brain imaging techniques such as CT, computerized tomography, MRI, magnetic resonance imaging, PET, positron emission tomography, and SPECT, single-photon emission computed tomography, have come into practice to study electrophysiological changes during different behaviors. 10.3 Method to assess population density. Population is the number of organisms of the same species that live in a particular geographic area at the same time. The population density may be defined as simply the number of individual organisms per unit area. Different species exist at different densities in their environments, and the same species may be able to achieve one density in one environment and different in another environment. There are 5 main methods for counting wildlife. These are total counts, sample counts, index counts, quadrant method, and mark recapture method. The choice of how to do the count, whether on foot, from a vehicle, or from an aircraft, will depend on the species to be counted, the size and relief of the area, the resources available, and the objective of the count. 10.3.1 Total count. A total count aims to count all the animals in a specific area. This area is called the census unit. It might be a national park, sanctuary, or forest patch. Because of undercounting, it's unlikely all the wildlife will be seen. Total counts can only provide a minimum estimate of the total population size. Total counts should be used only when the wildlife area is relatively small, under 10 square kilometers, and completely fenced, which means that no animals can enter or leave. For example, it is possible to do a total count of the animals in Tal Chapar Sanctuary of Sujagar Tahsil of Churu District, Rajasthan. A single species is being counted in a restricted area. For example, a total count of the king vulture, Sarcogyps calvus, in Jodhpur District, Rajasthan. Other than in small or restricted areas, total counts are rarely used because they only provide a minimum estimate, and the level of precision cannot be measured, and they are much more costly than sample counts. 10.3.2 Line transect method or sample count. For animals which are small or distribute over large area, samples of populations have to be counted to get an estimate of population. Sample count aims to estimate the numbers of animals in the total area within the census units from the number counted in a smaller area. Sample unit. Sample counts make two important assumptions. All the animals in the sample area or unit are seen and accurately counted. Animals are spread evenly throughout the whole wildlife area or census unit for which the population is being estimated. The extent to which these assumptions are valid is at the root of all the problems associated with counting wildlife. It is unlikely that all the animals in the sample area will be seen and counted, or that animals will be evenly distributed throughout an area. For example, we know that animals naturally congregate in areas of good habitat and where there is water. So careful planning has to take this uneven distribution into account if accurate and precise results are to be obtained. A road strip line transect count is the most common way of ground-based sampling. Road strip counts are usually used in smaller wildlife areas, 100 to 1,000 square kilometers. In a road strip count, a vehicle is driven along a selected network of roads in the wildlife area under survey. Observers in the back of the vehicle count all the animals seen and measure their perpendicular distance from the road with a rangefinder and an angliometer. The sample area is calculated from the average distance that animals are seen from the vehicle and the total distance traveled. If the size of the area is known, then the total population of wild animals in the area can be estimated. Figure 93. Line transect method for population census. An example of a road strip count is given in Figure 93 for counting of black bucks. The total wildlife area equals 270 square kilometers. Distance traveled by observers equals 54 kilometers. Average distance of animals seen from the road equals 0.5 kilometers. Total area surveyed equals 54 kilometers x 0.5 kilometers equals 27 square kilometers. Total number of black bucks seen equals 21. Therefore, in 1 square kilometer, there are 21/27 black buck. Therefore, in 270 square kilometers, it is estimated that there are 21/27 x 270,210 black buck. The estimated total black buck population is 210. The advantages of using road strip counts from a vehicle are that they provide estimates for more species than an aerial survey because most species larger than a dog will be seen. The disadvantages of using a vehicle are that a well-established road system is needed, a road system that is not biased towards certain habitats is needed, an accurate measurement of the distance of each sighting from the road is needed, a vehicle is needed, making it relatively expensive, and analysis can be quite complex. Walk transects are used mostly on small wildlife areas, 10 to 50 square kilometers, like small forest, or where there is a strong level of community management, for example, in a sacred groves or community protected areas. 10.3.3 Index method. An index method aims, by using a standard approach, to produce an indirect measurement of the status of the population in the total area. For an index to provide useful management information, data for it must be collected repeatedly over a period of time using exactly the same method each time. 4 types of method or index are commonly used. 1. Index of abundance. It gives an indication of the status of an animal population based on the numbers of animals seen per unit of time or distance in a particular area over several seasons. Example: In Jodhpur district, there is transect in the Makia Safari Park area. Each month, except in May and June, as it is very hot, the chief wildlife officer and one other monitor walk from main entrance to Makia Fort. They leave in the early morning and follow exactly the same route. They count all the animals they see from the path. They have been doing this since 2015. The results are given in the table below. The chief wildlife officer has noticed that the average number of desert fox seen each year has increased from 20 in 2015 to 40 in 2018. From this, he thinks that the number of desert fox in the Makia Safari Park area might have doubled, and there are at least 40 desert fox in this area. However, because he did not know the distance of the animals from the transect, or how long the transect is, or how big the Makia Safari Park area is, he is unable to estimate the total number of desert fox in Makia Safari Park area. 2. Index of trophy quality. It gives an indication of the status of the population based on the annual average trophy size, horns, teeth, fur size of a given species in a particular area over several years. Example, suppose a researcher not only recorded the number of animals shot on the quota available for hunting, but also the weight in kilograms of both tusks from all the sport-hunted male elephant in African wildlife area. Since 2010, the average weight has decreased slightly. From this information, researcher concludes that the number of trophy male elephant in the African wildlife area might be declining. 3. Index of hunting effort. It gives an indication of the status of the population based on the average time taken to find and shoot an animal. If over several years it regularly takes longer to find and shoot a trophy animal, this might indicate a decline in the number of trophy animals available. For index of hunting success rate, it gives an indication of the status of the population based on the percentage of the allocated quota which is hunted over several years. If the entire quota is shot every year, this implies that there is no shortage of trophy class animals. If the quota is not fully used, this might mean that there is a shortage of trophy animals, and the problem needs further investigation. Index methods or indices are important for establishing trends in populations over time. As long as the method used to collect the data is consistent, indices are technically acceptable. The data from many indicators is relatively easy to collect, and the costs are relatively low. So, index methods are affordable for small wildlife committees and/or district administration. 10.3.4 Quadrant Method. A quadrant is a frame, usually a square, of known area used to isolate a subset of the population. This subset will comprise one sample. Quadrants come in various sizes and shapes, with the size selected determined by features of the organisms in the population to be sampled. The use of a quadrant is very simple. It is placed randomly in the sampling area, the habitat of the species of interest, and all the individuals within the quadrant are counted and/or measured. Once a plot has been established, the total number of individuals of each species can be counted to determine population densities and species composition. It is generally used for sampling sedentary organisms. 16.3.5 Mark Recapture Technique. Mark recapture methods are based on trapping, marking, and releasing a known number of marked animals into the population and, after giving the marked individuals an appropriate period of time to once again mix with the rest of the population, some individuals are again captured from the population and some of the individuals caught in the second period will be carrying marks recaptured and others will not. If we assume that the ratio of marked to sampled individuals in the second sample n/r represents the ratio for the entire population n/m, we can compute an estimate of the population using the following relationships. The only variable that we do not know in this relationship is n. We can solve for n by rearranging the equation as follows. For example, in a census for a lake fish, 10 fishes are caught from a lake, marked with red-colored tags, and released back. A week later again, catch 15 fishes are caught, out of which 5 were previously tagged. If there's no immigration from that lake, which is quite likely, in a closed system, and it is assumed there have been no births or deaths of fish, then the total population size is given by rearranging the equation. Total population size, where n equals total population, m equals number of marked individuals in first catch, n equals total number of second catch, r equals number of marked individuals in second catch recapture. Using our data, thus, the lake has a total population size of 30 fishes. However, the mark recapture technique can have some limitations. Some animals that have been marked may learn to avoid the traps. In that case, recapture rates will be low, resulting in an overestimate of population size. For example, instead of 5 tagged fish out of 15 recaptured fish, there are only 2 tagged fish. Now our population size estimate is 150 divided by 2 is equal to 75, a dramatic increase in our population size estimate. Conversely, if there are higher numbers of recaptures, this would result in an underestimate of the population size. Tag types may vary with study organism. Larger mammals may receive ear tags, while smaller mammals and birds receive bands or rings. Insects may be painted in various colors. Dusting with fluorescent pigments may permit detection of marked recaptures by the use of ultraviolet light. For some larger terrestrial or marine species, captured animals can be fitted with a radio collar, a collar fitted with a small radio transmitter. That enables researchers to follow the animals telemetrically using an antennal tracking device. Their home ranges can be determined and population estimates developed based on the area of available habitat. For many species with valuable pelts, we can track population densities through time by examining pelt records taken from trading stations. We can also estimate relative population density by examining catch per unit effort, which is especially valuable in commercial fisheries. Although we can't easily count the number of fish in an area of ocean, we can count the number caught, say, per 100 hours of searching. 10.4 Ethical Guidelines for Animal Behavior. The use of animals in behavioral research and teaching does, however, raise important ethical issues. While many behavioral studies are non-invasive and involve only observations of animals in their natural habitat, some research questions cannot be addressed without manipulation of animals in the laboratory or field. In all research that involves animals, it is imperative to minimize individual stress and suffering. Research on animals is monitored and must be approved by institutional and sometimes government animal care and use committees. The 3 Rs replacement, reduction, and refinement are often used as guidelines. Replacement, an ethical guideline that encourages the use of computer modeling, videotapes, or other approaches in place of actual animals. Reduction, an ethical guideline that promotes limiting the number of animals subject to disturbance in research or teaching. Refinement, an ethical guideline that involves improving procedures and techniques to minimize pain and stress for animals. Researchers must consider the possibility of replacing animals with computer models, reducing the number of animals required, and refining their techniques to minimize stress and disturbance to the animals used. 10.5 Scientific Literacy. How do we determine what is based on valid research and what is just personal opinion? The ability to evaluate scientific information critically and ascertain its validity is called scientific literacy. Scientific literacy includes understanding how hypothesis testing drives the scientific process as well as peer review, in which scientists determine whether research is clear, valid, and important enough to be published in the primary literature. Animal behavior research is conducted by investigators and research students at college, universities, zoological parks, government and private research institutions, and conservation organizations. After conducting research, scientists write papers and submit them to scientific journals to report their findings. These journals, known as the primary literature, are the primary or original source of scientific information. Papers in scientific journals follow a consistent, systematic format. If the results of scientific research are of interest to the public, they may also be reported in newspapers, magazines, and books, as well as on television and over the internet. Reports like these are called the secondary literature because the information has been presented before and often does not have the same validity as the primary literature. 10.6 Proximate and Ultimate Cause of Animal Behavior. What kinds of research questions can we ask about animal behavior? The study of behavior critically depends on our ability to phrase our general inquiry as a series of questions that can be answered with standard scientific approaches that are amenable to experimental probes, and which lead to an explicit and unambiguous interpretation of its results. Nico Tinbergen's legacy as an exceptionally gifted experimentalist include the phrasing of 4 different types of questions that can be asked of any animal behavior, now simply referred to as Tinbergen's 4 aims or 4 why. As Tinbergen, 1963, pointed out, there are 4 different ways of answering the question why in biology. We can illustrate them in the context of the production of song by the zebra finch, a common songbird, as an example. Why do they do this? Answers at Tinbergen's 4 different levels might be as follows. 1. Causation/mechanism. What causes the behavior? What triggers the behavior? And what body parts, functions, and molecules are involved in carrying it out? Answers here are concerned with how the system operates in terms of underlying mechanism and organization. Example, singing is triggered in zebra finches by social cues, such as the proximity of a potential mate, as well as the appropriate hormonal state. The ability to produce songs is influenced by male hormones and occurs mainly in male birds. Songs are produced when air flows from air sacs in the bronchi through an organ called the syrinx. Certain parts of the brain control song production and are well developed in male zebra finches. 2. Development. How does the behavior develop? Is the behavior present early in life? Does it change over the course of the organism's lifetime? What experiences are necessary for its development? These kinds of answer are concerned with the way the system reflects its embryological, cultural, or other developmental influences. Example, young male zebra finches first listen to the songs of nearby males of their species, particularly their fathers. Then they start to practice singing. By adulthood, male zebra finches have learned to produce their own songs, which are unique but often have similarities to those of their fathers. Once a finch has perfected its song, the song remains fixed for life. 3. Function/adaptive value. How does the behavior affect fitness? How does the behavior affect an organism's chances of survival and reproduction? Answers at this level are concerned with what the system is designed to do, i.e. its role in the life of the organism. Example, singing helps male zebra finches attract mates, increasing the chances that they will reproduce. Singing is part of an elaborate courtship ritual that entices the female to choose the male. For phylogeny/evolution, how did the behavior evolve? How does the behavior compare to those of related species? Why might it have evolved as it did? Here we are concerned with the ancestral selection pressures and phylogenetic pathways that have shaped and constrained the system. Example, almost all species of birds can make vocal sounds, but only those in the suborder Passerii are songbirds. Relative to the zebra finch, other songbird species differ in the timing of their listening and practicing phases, the plasticity of song over their lifetimes, the extent to which the song is similar among. Tinbergen's 4 questions provide a framework for the study of behavior. Answers to questions 1 and 2 are often referred to as proximate explanations, and answers to questions 3 and 4 are known as ultimate explanations. 1. Proximate causation deals with questions like how behavior is performed, that is, how the animal's senses, nerve networks, or internal state provide a physiological basis for the behavior. These explanations often incorporate studies of genetics, sensory systems, neurons, hormones, and learning. 2. Ultimate causation inquiry are about adaptive/evolutionary value of behavior. To study the ultimate cause of a behavior, we would attempt to determine how it influenced the animal's survival or reproductive success. For example, a male songbird may sing during the breeding season because it has a level of the steroid sex hormone, testosterone, which binds to hormone receptors in the brain and triggers the production of song. This would be the V proximate cause of the male bird's song. But the male sings to defend territory from other males and to attract a female to reproduce. This is the ultimate or evolutionary explanation for the male's vocalization.